Leakey had to pass a language exam. And so they searched the whole world for an expert in the language he wanted to pass it in. And the only person they could find was Leakey.
The imaging of the salamander eggs would be a good thing to study because I could show you my PowerPoint on it.
Yeah.
It's at some point I can show it to you sometime. I was going to pair it with the sucrose gradient and watch the salamander eggs rise in the sucrose gradient and take images of my sample salamander eggs as that was happening. And then you could see the inside, what the inside's doing while it's becoming less dense in the sucrose column. So it's a fairly simple experiment. We have all of the pieces that you need. Like I have the sucrose gradient.
Yeah.
So I thought it would be an amazing experiment, publishable, of course. And I want to go do that instead of tensegrity. Although I'm keeping on with the tensegrity. If anybody's interested, there's a course in it in Italy, but you can take it virtually and it's in English. And it's, let's see, called tensegrity system. It cut it off. September 16th to 20th. It's a week long, and it costs about $400 Canadian or $300 American for the course, for a week-long course. I'm taking it virtually from my living room.
Oh, you are?
Yes, if they approve me. I don't know.
They should take it, not you.
Oh, yeah. I'm going to tell Dr. Zhang this afternoon. There's his course that he can take. He's retiring, so he can take the course.
There you go.
I'm going to give it to him.
Yeah, welcome to the meeting. Looks like Vahid's here. Hello, Vahid.
Hello, everyone.
Hey. Well, Mehul and Morgan, Dick. So I don't know if anyone wanted to present anything or discuss anything before we go ahead.
Okay, I've been working on the problem that for a parser that we have been talking about in the past regarding those two cells that I'm trying to simulate to be able to, as a general sample or a use case study for other cell models in C. elegans, I've been trying to simulate those two cells, and I've been working with PathRate and actually the parser that some other projects in a recent study they have published some data model for two sample models for two cells in C. elegans. So I'm trying to simulate those two cells in Neuronal and LEMS, which are actually the basics of our simulations in OpenWorld. I could somehow simulate, try to understand what is the problem, because Patrick was working on it in the past, but I'm trying to solve some problems that we have, and maybe in the near future we could add those two simulation of the cells to the OpenWorld project, and after that we could maybe have a great platform for some cellular simulations that could be added as a DevoWorm or any developmental process is going to be simulated.
Yeah, that would be great. Yeah, I saw a little bit of an email chain here with Horig from OpenWorm. So, is that basically what you were describing?
Yeah. Actually, that's the basics.
Yeah, good. Well, that's great. Well, hopefully, yeah, I move along on that. And I don't know if you wanted to, if you want to share any results as you go along, you can do it in the meeting here. Or I guess we can chat on the side as well if you need. So.
For me or for?
Oh, I mean, well, for your project.
For sure. Okay. Actually, I was a little having some holidays and some relocation issues, so I'm on the roll from this week on, and I would be glad to present it in the future.
Yeah, it's good.
For sure.
Good luck. Yeah, it sounds good. Sounds like something. You. Yeah. Mehul, how are you?
Hi Bradley,
Hi.
I'm good. So actually, I'm currently I'm in Hyderabad, and next week I'm going to Delhi. So I actually told Bradley I'll be going to Japan for the conference in June. So I'm preparing visa and all next week. So I'm traveling to capital of India to get. So a lot of things are going on in that side. It will be my first offline conference to attend. So I'm very excited about it. I'll be look out for like computational biological networks as well in the conference. Anything happening there. So
yeah, what
I'll keep you updated on that.
Yep. Yeah, what conference is it?
It's WCCI IJCNN. So IJCNN is a conference, but like 3-4 conferences are happening together.
Okay.
Actually, I was taking that only if anything is related to development biology, but I didn't find anything related to it. But there are many brain-related keynote speakers and workshops are happening. So brain development and all those things. So I think Morgan will be interested in that.
Oh yeah, that sounds great. That's great. And oh, go ahead.
Yeah, nothing else. I'm just like into that stuff only. So, not doing anything cool right now.
Yeah. Well, that's good. Yeah, so thanks for that update. Yeah, your presentation last week was pretty good. Yeah, I'm looking forward to going over the proposals and seeing what we can get. And definitely want to work on that project. Yeah, so let's see. I have a special here, and I don't know if people realize it. Well, Dick probably is the only person who would know this or maybe remember this, but it's the 10th anniversary of DevoWorm. So this is
really?
Yeah, this week. So I'm gonna.
Hey, I got some cake to eat today.
Oh yeah.
Okay.
So yeah, I'm gonna.
Boy, we've been at it that long, huh?
Yeah, 10 years, 2014. So yeah, 10 years of computational developmental biology. So I'm gonna make us talk on this and put it on YouTube. Like it's gonna go over kind of the activities over the 10 years, a recap of things, all the different papers that we put out, all the different things that we've done. We've done Google Summer of Code for probably about maybe about 7 years now. So it's been a while. I think this will be the 7th year. And so we have a rich history there. We have a lot of presentations that we've done, a lot of publications, a lot of meetings where we've talked about things and gotten things off the ground. So yeah, I'll be putting a longer, probably about an hour-long talk on YouTube sometime this week. I have it almost ready, but I have to record it as well. So this is, we're doing this computational developmental biology. It's this open source participatory model. We have our different modes of doing things. So we have GitHub, YouTube, Hugging Face, the OpenWorm Foundation, the Orthogonal Research Lab, and then we use Jitsi as a way to do video conferencing. So,
Maybe,
go ahead.
maybe we should have Maybe we should have a meeting next week and let you have a practice.
Maybe, yeah.
Okay, then we can put in our 2 cents.
Yeah. So this is the first. So the way this started like in April 2014. So I think Dick actually got in touch with Stephen Larson or something like that, and
I don't remember.
okay, yeah. Well, Steve Larson, back then was all committed. He's very committed to OpenWorm, and he's been spending more time with his startup Metacell. But back then, he was managing like issues on OpenWorm, and so they were doing things with the worm simulation, and they're doing things with PyOpenWorm, which is just the Python version of OpenWorm. So there was this open issue in their GitHub repository. It was issue number 179, and this was actually something he opened in response to what we wanted to do. So we had a meeting with Steve Larson, and he talked about basically incorporating something into OpenWorm that was developmental. So there's this classic data set from Solston, and he actually sat there at his in the lab, and he drew out a lineage tree by hand. So he looked under the microscope, and he looked at C. elegans, the different cells, and he was in an embryo, and he was able to actually map out the different cells in the embryo. So and this was all done by hand. It was drawn. So if you see any sort of lineage tree, know that was done by hand. And back in the 70s, so it wasn't that long ago, actually. You'd think it would be a little bit higher tech back then, but so
54 years ago.
Yeah, which
You're
yeah,
showing your age.
yeah. I mean, historically, like it wasn't in 19th century or something. So we had an issue here that we wanted to build here. And there were a couple things. Identify the best data sources to begin working with. Write a simple Python script that pulls out the names of the cells and the cell division relationships and displays the console. Augment the script to dump the data into PyOpenWorm SQLite database. Augment PyOpenWorm to pull these data out from SQL database and construct an RDF graph, which is a data structure out of it. And then build accessor functions in PyOpenWorm. So we actually didn't actually go in this direction, but this issue was sort of the beginning part. What ended up happening was we ended up kind of diverging from like the OpenWorm Foundation a bit and doing our own thing, which is fine. But that's what happens a lot of times in open source. You get forks. We took a fork off of this. I don't know whatever happened to this issue, but it's a good, when you want to start something like this, it's good to lay it out in an issue and then see where people want to go with it. So, we actually, that's what we did with DevoWorm, is we've kind of forked some of the work we're doing in, and I'll show this in the presentation with like kind of our machine learning applications, and kind of went off in a forked direction instead of like just assigning in-house issues. So this was our first meeting, April 11th. We used Google Hangouts, so that actually was pretty long ago, because Google Hangouts is no longer a thing. But that's how long ago it was. And still, Steve Larson is here. Myself and Dick were in the meeting, and then we expanded our group over time to include new people. Some people left, some people came in. There have been a lot of people who have gone, come and gone, and that's the nature of this type of group. You get people visit for a while and then get what they need and leave, and then some people stick around for a while, and so that's. But this is where it started on April 11th, 2014. So we made I think I made the first public announcement on my personal blog on June 3rd, 2014. That was in the summer. This is a presentation I think we did with the OpenWorm Foundation. So back then they used to have talks, public talks that they did, where they would have some of the sub projects give talks on their progress, and it was much more interactive than it is now. But that was sort of the nature of the project at the time, and so we did a couple on DevoWorm, and so we took that Solston data set and ran with it. We had other data sets as well, like cell tracking data and other types of data, and so that really built the early days of the project. Now, we had also talked about things like finite element analysis and cellular automata and things like that. And we did actually follow up on the cellular automata stuff. We did some work there. We could do more. We never really did anything with the finite element analysis stuff, but we did, do some things with the lineage tree data, with some of the cell tracking data. So, they're always really based on the secondary data sets that other people have collected. And that's really what OpenWorm's done as well. They've done a lot of things with secondary data. And in C. elegans, you have a wealth of that kind of information. People have collected a lot of data, but not really done anything like theoretical with it, or, the modeling is, there's a lot of modeling work that should be done that people aren't doing for various reasons. So that's the public announcement. By then, we had Mark Watts, who was a senior contributor at OpenWorm. I think he quit OpenWorm a while back, but he worked on a lot of stuff with respect to like modeling, like building data structures and working in Python and things like that. And then Steve McGrew, who sadly has passed away by now, he also joined the group, and he was kind of one of these people who, had a lot of talents. And so he was really interested in the project, and he joined our group by that point as well. So there's a lot of stuff that came after that. We had an open house in 2016. We started Google Summer of Code in 2017. We got some papers on 2016. So there were a lot of things going on, and throughout it all, I think we've kind of maintained I think we've maintained things pretty well. And then our first publication, which was October 3, 2014, this is on the bioRxiv. This is differentiation waves and computation. It's kind of like we're putting our flag down and marking some territory. So this is, kind of the first thing we did. This is where we proposed a 4D model for the C. elegans lineage tree using the RDF framework, which is this tabular data structure that sort of, ties together a lot of the lineage tree data with some other data that exists for C. elegans development. So I haven't gone back to that paper and read it again, but I remember that was the first thing we did. And so I had, after that we did other presentations before that, but so
Bradley.
Yeah.
The amusing thing is it took for 24 years to get something like this going. Because in my 1999 book, Hierarchical Genome, you'll find a description of cyberworm.
Oh, right.
Yeah, cyberworm was, in a sense, its predecessor, which I couldn't get off the ground. I had somebody interested in it, then faded away.
Yeah.
Okay, so it took meeting you to get it going.
Yeah. What's interesting is there have been a lot of people proposing that you do things to C. elegans. Like there are a lot of, I went through like the web, and I knew this was happening because I've seen papers on it, but we did this paper a couple weeks ago on MetaWorm, which was an attempt to take some, different tools and apply them to analyzing C. elegans. And it turns out there have been a lot of attempts at this over the years. Not so much development, but like modeling C. elegans. And so, there was the Perfect C. elegans project.
That's a question.
Yeah.
Did Tom Porteous create a robot that was like C. elegans?
Yeah, he did. Well, yeah, that was something that. So there was an open house at the Royal Society in 2018, and that was OpenWorm. OpenWorm sponsored that, and he actually developed some robots. At that time, 2017, 2018, he was really deep into working on robots. And they actually did develop a robot that was, it was like it wasn't like the size of C. elegans, it was scaled up so you can interact with it on the tabletop. But it uses the algorithm that they have for movement. So you'd put in the algorithm, you could run this robot, and it would have like a lot of the same behaviors as the C. elegans. And there's a lot there to do because there were a couple of people who were doing work. They were contributors. Well, this was back in 2018, where they were looking at soft robotics, where you have soft bodies. So, C. elegans, of course, is a soft body like every other organism. And, you could build robots with, metal or plastic, but it's like you can use more exotic soft materials to get sort of compliance you need to make some of the, movements like omega turns and things like that. And so they were working on that. I don't know if that went any farther because people were working on it for a while and then it stopped. And I know they were talking about selling a kit. I don't know if that's still around, but yeah, it's definitely been explored, and it's an interesting kind of model because you can test hypotheses about movement, about biophysics that you couldn't do just by doing things in a computer. Like the whole thing in a computational simulation. You can actually take the movement program, you can put it into a body of some type, and it's interacting with the world. And so it's C. elegans isn't moving around a tabletop, but you can do things that are you can actually see things. They were using it for education. That was the initial application domain, but you could easily use that for research. I mean, it's not. You could probably learn quite a few things from that. So that was the first publication. Yeah, so that was that's. I'll have a full presentation at pretty soon. I'm going to finish it off, and then we'll see. We'll kind of go over it and see how much progress has been made because it's pretty surprising to me, anyways. Going back over it and seeing all the things that we've done. So.
And how far we have to go.
Oh yeah. There are a lot of things there. Yeah, there's always more to do, I guess. But yeah. That's great.
Okay, well, maybe we can get a practice session next week.
Yeah. So we had a couple things here. So there's this interesting paper that came out, I think it was like a couple weeks ago, maybe. I can't tell where it was, maybe March 22nd or so. So it was late last month. And it's in the PNAS, it's a research article in PNAS. And this is cell reprogramming designed by transfer learning, a functional transcriptional network. So this is cell reprogramming, which is a little bit different than what we talk about in C. elegans. It's where people are they're trying to transform cells from like something like a skin cell to a stem cell, or a stem cell to a neuron or something like that. And what they'd usually do is they use transcription factors to drive that process. So they introduce transcription factors into the cell, they transfect them in, and then they can get the cell to transform in some way. And so the problem with that technique is it's great because you can create cell populations in the lab that you couldn't create otherwise. Sometimes you can't get access to certain cell types. But the problem is it's somewhat inefficient. And people have not, I don't know if they've not been honest about the efficiency, but it's not something that's, easily, it's not easy to do in the lab. And, people want to use this eventually for therapies, for cell transplantation or things like that, but also for research and having like a stable model. So that's where they're coming from. This Adelson Motter is well known in network science for some of the work he's done with networks, but also biophysics. And then Thomas Wytock is an author on this. So this is so they're talking about cell programming design, which is where you're designing sort of a protocol for the cell reprogramming, and then transfer learning of functional transcriptional networks. This is where you're trying to learn, these transcriptional networks and how they can be manipulated using key transcription factors. So if you upregulate a certain transcription factor, you'll push the cell towards a certain fate. And so, if you go back to the work of Yamanaka and the 4 transcription factors, that's the work that won a Nobel Prize. They basically took they did a bunch of screens of stem cells, and they found that there are 4 genes that control what they call stemness in cells. And so they applied those 4 transcription factors to a cell. They put them in the cell. They over-regulated those genes, and they were able to push the cell into a new fate, which means that there are these transcription factors that sort of control the process and ultimately control sort of the nature of these regulatory network, these transcriptional networks. And it's not common. You have to do a lot of screening, but you can find genes of large effect that do this. So that's kind of what they're trying to do, and that's just for stem cells. If you want to transform, say, stem cells to neurons, you have to have a whole different set of transcription factors, and then it gets a little tricky because there are a lot of different types of neurons, and there are a lot of different ways to do it. So this is where they're coming from on this. So if we look over here at the significance statement of this paper, over here, you'll see that they say the lack of genome-wide mathematical models for the gene regulatory network complicates the application of control theory to manipulate cell behavior in humans. So what they're trying to do is they're trying to develop some sort of genome-wide mathematical model. And this is to look at gene regulatory networks. So, you want to build a mathematical model of a gene regulatory network. And typically this is some sort of model where you have genes that interact in some way. So sometimes, it's usually sort of causal. So you have one gene that maybe upregulates another gene, maybe another gene that downregulates another gene, and so forth. And so you can see these, you can draw these huge networks on the board where one gene affects another gene. But the thing is that, they're not just simply one thing affects another thing. There are a lot of intermediate things going on. There's a lot of mixed valence. So, for example, one gene might upregulate something, or it might, cause another thing to be upregulated, but there might be other genes that downregulate that thing. So it's hard to know which thing has the most effect if you manipulate it in some way, or if you overexpress something in some way. How much of an effect does that have? So that's what they're trying to do, build a mathematical model of this. So they're trying to be able to do this by developing a transfer learning approach that leverages genome-wide transcriptomic profiles. So they're using data from transcription. So they're looking at genes that are upregulated or downregulated in the cells, and you can get these genome-wide transcriptional profiles, and you can say if something is upregulated or downregulated in a cell. And so those profiles, you can use those to sort of learn what constitutes a cell's, what kinds of genes a cell is expressing, or how much if they're genes that are really highly expressed, that's usually a signal that they're involved in some sort of function, or something like that. So that's what they're trying to do here. So they're trying to build these cell type attractors and perturbation responses. So they're trying to figure out also not only the identity and the function, but what happens when you perturb a cell. What are the things that change? What are the things that need to change? If you're going from one state to another state, what are these kind of things? So that's where they're kind of coming from. They're using this transfer learning approach. These responses are used to predict a combinatorial perturbation that minimizes the transcriptional difference between an initial cell and a target cell type. So what they're doing is they're building this model where you have, let's say, a stem cell on the one hand and a neuron on the other hand, and you want to find the transcriptional profile for each cell type. And then you want to find the difference between those two cell types in terms of their transcriptional profile. So which genes, when you look at their transcriptional profile, change the most. So some of them are going to change a lot. Some of them are going to change a little bit. And some of them are going to maybe change a little bit but be negligible, and so forth. So we want to identify the things that are sort of, different, but also the same. And then we want to be able to predict perturbations that minimize that difference. So we can actually, once we do that, then we can build a model with a regulatory network. We can bring that network to the target cell type basin of attraction. We anticipate that this approach will enable the rapid identification of potential targets for treatment of complex diseases. So this is where I was saying that they want to do things in the clinic with cellular reprogramming, but there are a lot of barriers to that in terms of knowing how the cells are going to behave, especially in vivo. And so it's very hard to know these things, and that's why I think we need mathematical models for that. While also providing insight into how the dynamics of gene regulatory networks affect phenotype. So this is something else we don't really know a lot about, is how these networks affect the phenotype. How do you go from a network that has, all these genes fluctuating up and down? How do those translate into a phenotype? And obviously you need to go through several more steps. You need to go through translation. You need to go through post-translational modifications. You need to go through protein construction and protein folding and things like that to get a phenotype. But this is a good step in the right direction here. So in the paper, they talk about synthetic biology and some of those applications. And they just basically talk about, some of these protocols to control cell behavior that people are starting to develop with respect to synthetic biology. So if you're going to do synthetic biology, you need to be able to control your cells and know where, if you make a manipulation, what the effect of that manipulation will be. You need to be able to predict that. And so, it's hard to do this with brute force trial and error. So a lot of times the way we do these kind of assays of sort of perturbation assays is to do like knockout experiments where you knock out different genes and you look at their effect. But that can be very time consuming. Same thing goes with like screens. So if you do a genetic screen, you look at maybe you test for a bunch of different transcription factors at once, and you try to isolate the ones that are that change the most, and you do more tests where you try to manipulate its expression to see, under what conditions it's having a large effect. Those things are all very time consuming, and so it would be nice to have a mathematical model that would streamline that process. And so they talk about control theory being, an engineering discipline, and how in biology you can use control theory, but you need to approach it in a different way. So unlike engineered systems in which control theory provides an equation-based framework to design interventions, so in control theory and engineering, we have equations that predict the outcomes of systems, that we can control something like an industrial process. We can control, speed of something or the trajectory of something. But, in biological systems, we don't really have those descriptions. We don't have like equations of motion for transcription or for, cell behaviors. We'd like to be there, but we're not there yet. So, one of the problems with building such models is that it's hard to constrain the numbers of actual degrees of freedom in terms of genes to describe a cell. So when you look at genes, you have a lot of genes. They're being expressed in different ways. Those are basically akin to degrees of freedom. So you have this massive number of degrees of freedom that you need to constrain as you try to, do engineering or build a mathematical model. And so it's very hard to control those because you simply don't know what, if you hold something constant, whether that's the thing you shouldn't be holding constant. So it's very hard to do this in an educated way. I mean, you can take educated guesses, but it's like sticking your finger in a dam. It's just not feasible without really some good models. So the features presented a challenge given that underactuated control is onerous even in physical systems that admit a closed form mathematical description. So this is where they talk about underactuation, which is the way they view biological systems. And you could say that they're also underspecified, another way of saying this. And this is a problem, of course, in modeling where, we'd like to have the right level of specification. If things are overspecified, there's too much, there's maybe the system is too rigid. If it's underspecified, then the system isn't very predictive. And so this is something we can't really find the, middle ground on. So this is a problem. As a result, the biological control problem is often relaxed to steering between natively stable states. So, in other words, we know kind of what the stable states are maybe, which is not really biology. So we know, we know maybe what an idealized stem cell is. We know what our model stem cells are. We know what like the wild type is that we work on in the lab. But if we look at like biological diversity, we know that there are a lot of these unstable states that are also very useful and interesting. And especially when you move between different cell states. You move between a stem cell of some type and a neuron of some type. Well, that, those are at least nominally stable states. But you actually go through a series of unstable states. Furthermore, a stem cell could be sort of an, kind of an off stem cell or maybe an unstable type stem cell. And you could end up with an unstable type neuron. And you don't really want to be in there, or maybe you do. It depends on what your end goal is. The point is that we know how to go from the idealized stem cell to the idealized neuron, but we don't know a lot about how to control the system to move to maybe unusual states or unstable states. So they talk about their goal here is to design a general data-driven control approach tailored to these aspects of the data. And their approach contrasts with related control theoretic formulations in the literature, which cannot be easily deployed as they usually require targeted experiments to design the controller, temporally rich data, individually resolved system trajectories, and the availability of microscopic agent-based models. So these are citations 18 through 22. So 18 is this paper, Danny Bassett's an author on it, Data-driven control of complex networks. So this is this area of network control where they're looking for certain nodes that control the network. That's an interesting area of research. 19 is generalizing Koopman theory to allow for inputs and control. This is dynamical systems theory, but this is also something that is not, this is Steve Brunton is an author on this. So again, it's generalized sort of control theory, but it's maybe something we can use in biology. This paper here, model-free control of dynamical systems with deep reservoir computing. So reservoir computing, for the machine learning people, it's an up-and-coming technique that people are using to model biological systems, connectomes, things like that. I've seen a number of talks where they kind of, highlight reservoir computing as an up-and-coming approach. I've seen talks where they've done, things where they've modeled connectomes, where they've modeled the inside of cells. And basically what a reservoir computer is it's sort of a randomized neural network. And the idea is you run things through a reservoir computer, and it has a reservoir of nodes and weights. So it puts together the answer. It emerges from this sort of reservoir, and then it gives an answer. And it's something that is, at least to me, it's a black box. I'm not quite sure I understand how to implement it, but it's something that is, people are there's a lot of promise there. This is 21. This is Danny Bess. It's also an author on this. Teaching recurrent neural networks to infer global temporal structure from local examples. So this is where you're trying to get predict global temporal structure. Again, this is just generalizing things. And then this 22 is data-driven control of agent-based models, an equation variable-free machine learning approach. So this is again, working now with agent-based models and using data to control those and control theory and all that. So there's a lot of stuff in here, a lot of different topics. Network control, of course, reservoir computing makes an appearance, dynamical systems, and even some other types of mathematical approaches like agent-based modeling. So there's a lot of math and a lot of computation here that they bring to this paper. It's somewhat inscrutable in parts, I guess. I didn't really go through the details, but this is the kind of work that people are doing, especially like in biophysics on these topics. So I don't know if anyone had any questions. Let's see, we have some things in the chat. Oh yeah, Mehul said there is a student working in reservoir computing in my lab. Yeah, it's a topic people are picking up on it, but it's not. I don't know how far we are along. It's not as common as neural networks and things like that.
What is reservoir computing?
So it's
in general?
Yeah, so it's kind of like it's based on neural networks, and you have this this model that's a reservoir, and it has like a lot of nodes in it and a lot of edges that are unorganized. So it's like I don't want to call it. There's another technique that uses that terminology, but basically you feed data into it, and it makes connections based on what's coming into it. So it's learning basically the network topology from the data coming in. And so, it sounds like, your typical neural network, but it actually has like this reservoir where you're just kind of drawing from what's available, the computational resources that are available, and it's exporting an answer. And so, I guess it doesn't have as much. I don't know what the advantages of it are, but yeah. So it's that kind of a thing. It's sort of an unsupervised model, I guess. But it's related to neural networks, but not quite the same thing. And people are making a lot of connections between that and complex networks, where we have networks that are based on some sort of data set where you're finding the connections, you're finding like hubs of nodes that are hubs, and nodes that control processes. So in the C. elegans connectome, for example, they're hubs that control different parts of the connectome. So a lot of nodes that are downstream of that hub have to go through the hub to get to another part of the connectome. So it controls that part of the connectome. And so I think in reservoir computing, the neural networks can actually resemble that kind of hierarchical control. I'm not really sure how people are implementing it, though. There are a lot of different ways. It's kind of one of these newer techniques where there are no rules. Well, I guess there's some rules, but it certainly isn't as established as some of the neural network techniques. Okay.
Oh, thanks. That's quite involved.
Yeah, I know. So yeah.
I know a bit about control theory, but just sort of on a practical level.
Yeah. You can build pretty involved models, of course.
It's they're definitely being used. I'm not exactly sure either, but there's some connection with random matrix theory.
Yeah.
And it is used in kind of specialized high performance computing scenarios too.
Right.
Like it definitely has a connection to recurrent neural networks, but there's definitely some
because again, like the light-on folks in Paris are using this with their photonic computing, and then we saw it with the brain gnome, or I forget what that awful name was, the organoids, the organoid doing reservoir computing to do like sound sample identification.
Okay.
There was a recent paper, if you remember that.
I don't remember.
Anyway, but again, it seemed like they were kind of using the organoid to do this random reconfiguration of the inputs,
Right.
and then having the original and this new configuration was part of the processing stream.
Yeah.
Which again, I'd really like to. I'd like to know more, but yeah, it has some useful properties, but they seem to be quite specific. Certainly not as like as kind of wide purpose as general neural networks.
Right. Well, yeah, I mean, like they have that aspect where you're building up from random, so you're not like, it's almost like a complex network where you compare the topology to a random topology, and you say, what is the structure of this thing? Given the random topology is like kind of the ground state. In a reservoir computer, you have this random set of connections, and you're saying, what is the structure that the data is introducing to this? So it's kind of like you're building the structure from that. Yeah. So almost like a dynamical, you're throwing a dynamical system at it. Yeah.
Yeah. It's like I see national labs using it, and like not a lot of other groups, and I don't know what that means.
Well, it probably means there's a lot of computational power that's required to really run them, but.
Well, yeah, it does. I mean, again, like I'm curious what the light-on folks would say, because it's like I do feel like it's like you've got to do this at a scale that makes it useful, and this kind of light parallelism allows you to do that, to somehow exploit this random projection. But again, yeah, love to know more.
Yeah.
Love to have somebody who understands this explain.
Yeah, I thought that they had some pretty good insights about control theory, and not being an expert at control theory, but I think that's my caveat. But I mean, the idea that if you have in biology, and we've actually talked about in the other group, we've of course talked about the cyberneticists and how they kind of viewed biological control, and it wasn't really it wasn't as formal of a theory as control theory is now. But there's special dispensations you have to make for biology, as opposed to like controlling a dam or controlling a nuclear power plant. That you have these parameters that are, it's a really underspecified system. So we don't know like a lot of things about like transcription, and sometimes those are just stochastic variables or parameters. So you can't say like this is we can just learn more about it because it's just inherently stochastic. So that's something you have to, but we don't really know what things are which. So it's very underspecified. So that's an interesting point that, in the sense that you can actually apply control theory to something like that. How would you do it?
Yeah. Well, I'm curious how this relates to this Regulon paper. I put it in the DevoWorm orthogonal. But I'll put it in the DevoWorm Slack too.
Yeah.
And I'm curious. This gene regulatory network, framework that, or analysis framework that they put together that seems related.
Yeah, another thing about gene regulatory networks is that like a lot of people have like kind of run them, like they kind of design them and they say this is how it works, but it's hard to simulate them and it's hard to apply them to like real world data. So I think people are making progress on it, but like traditionally it's been very hard to get like good solid answers out of them. So hopefully, some of the techniques people are using.
Again, this is why we need a lot of good organoid, publicly available organoid work.
Yeah. So a few words on genetic regulatory networks. So as I mentioned in the meeting, a genetic regulatory network is something like this, where you have individual genes and the genes are expressed. So usually you have this model where you have some sort of promoter at the front. You have a gene, which is the square, one of these rectangles, and then you have this arrow that comes up, which denotes the expression product that comes out of it. So I'm just drawing these bubbles kind of as a cheeky cartoon of it. But these genes are also connected. So this gene up here is connected to this gene down here, and it's a causal relationship. So this gene A will cause gene B to be upregulated, and gene B will express something. So A precedes B, but A triggers B to be expressed. Now, you can also have another gene here, C, which is here, and you can have products. And you can see that C is induced by B. So we have A inducing B, B inducing C, and you have this circuit that is sequential gene expression. But it's not so simple because, first of all, we can have inhibitory relationships as well. So we can have an inhibitory relationship between B and C, and that will shut down expression of C. So if A is upregulated, if it's expressed, then it will trigger B to be expressed as well. But the expression of B then inhibits the expression of C. So we can have our diagram here where A contributes to the expression of B. B gets expressed, but then it inhibits the expression of C. And so A is going up, B is going up, and C is going down. And so a lot of times in gene regulatory network models, you can get real data to plug in here, gene expression data. And you can say that, A, B, and C are related, and you can see the sort of polarity of the model as you work through it to see what's being inhibited and what's being sort of encouraged or excited by downstream or upstream components. But it's even less simple than that, because you can actually have other genes that are affecting things. So a lot of times gene expression networks or gene regulatory networks are, kind of extracted from real data. They're extracted from the literature on studying the effects of single genes. And sometimes people will build models that just are kind of like maybe a few genes and maybe the genes of largest effect. But you have a lot of other genes here. You might have A prime, you might have B prime, and you might have a lot of minor players that actually maybe aren't expressed, but they are still contributing, expressed at least in terms of their own function, but they still play a role in the expression of B. So maybe B prime is inhibitory. So if B gets expressed, which isn't very often, it inhibits B. So A can contribute a signal, A prime can contribute a signal, and B prime all contribute signals. The signals remain A prime or facilitatory, and the signal from B prime is inhibitory. So you can actually add those together in a model where you consider all the effects collectively. And of course, a lot of times different genes will have different contextual effects. So for example, if you're in a certain environment, B prime might be expressed and inhibit B in the face of the expression of A. A might also get shut down in other environments which affect the expression of B, and so on and so forth. So these can become very complex and very detailed very quickly. Now we also talked about reservoir computers. And so to be very graphical about this, reservoir computer is in a reservoir. It's sort of a black box model. And so within that black box, or at least coming into the black box, we have an input. And we have an output coming out of the black box. so we have an input and an output. And this input and output then are shaped by what's in the black box. So the black box contains this randomized network. So this is what Morgan was referring to when he talked about the connections to random matrix theory, is that you have basically this randomized network in here, and it's basically represented by a randomized matrix. Now, what happens when you get an input of data is that it shapes this network so that it takes on some sort of topology that actually can process this information and provide some feedback. So this is something that, can be shaped from a random network to something that's non-random. And so we think of our, weight matrices in our neural networks as being non-random. It's selected by, the data, and it's shaped by the data. And so we have this transition from random to non-random. And this is what we see in a reservoir computer, is we have this reservoir of nodes and this reservoir of edges, and it gets shaped by the data, and it gives us an answer, which is, of course, the answer that we might expect from, a neural network. Now, there's also some interesting connections in the paper with network controllability or network control, and sort of these changing topologies. And so there's actually a paper that just came out. It was from a group of people, including Michael Levin, where they're looking at genetic regulatory networks, which we talked about earlier, and their competencies. And so their competencies are really based on this notion of network control and changing topologies. So if you have a network that has a number of components, and they have a number of effect sizes. So say, for example, this A is feeding into B. The expression of A is, or the expression of B is contingent on A, and so forth. But they have different effect sizes, so there are other things feeding into B, and A only has a small effect on the expression of B, although it does contribute. We can find the nodes that have the biggest effect, and the things that affect downstream things. So we have, in a complex network, we have hubs like this, and they control all of these downstream nodes. These nodes are sort of dependent on this hub for connection to the rest of the network, to the wider network. And so this hub is said to be a control node. And you see these control nodes in the connectome, where you have different neurons that have a large effect on the topology. It might be on movement or controlling some other function. And so that means that there are a lot of nodes that are feeding into this hub. And if the hub is removed, or if it changes its function, it can affect large portions of the network. And so there's some interesting connections between connectomics and reservoir computers, because a lot of people think that you can use connectomics to or you can use reservoir computers to sort of find connectome, different types of functional connectomes. So one other thing I'd like to go over before we end for today, and that is this thing we've had. So Dick actually put together a paper in 1994. This is with Ryan Drum, and I think this is in response to something that came up in a meeting a long time ago, but I can't remember the exact context to this. But this is on branching morphogenesis. So this is one of Dick's papers. This is the chemical basis of diatom morphogenesis. So in the group, we've worked on diatoms and single cell morphogenesis in diatoms. So these are diatoms, one variation of diatoms, where you have this structure. It's a single cell, or I guess it's considered a single cell organism. Some diatoms come in colonies, but you have these cells that have this structure, and they kind of grow from, throughout their lifespan, and they have these accretions, and they have this structure here, this branching structure. And so this is an example of a diatom they're kind of talking about in this paper. And so this is the chemical basis of diatom morphogenesis, which of course is a takeoff on chemical morphogenesis of Turing and some of the things that he talked about in his famous work. So this is just to talk about this abstract. An examination of the artificial diatoms shows that purely chemical and physical considerations will account for the varieties of pattern we notice in natural diatoms. And their living structure appears only to provide the conditions under which this silicous precipitation takes place. So they have they're silicate organisms. They have this precipitation that contributes to the growth of the cells and the shells. And so there's this morphogenesis that takes place via silica deposition. According to the ordinary laws of chemical action and molecular coalescence, I have tried to make the subject more intelligible. To that, I fear very numerous classes of microscopists who have not paid attention to Schultz's artificial diatoms the attention they deserve. So, I don't know, Dick, could you provide more context to this paper? Oh, are you there?
Yeah,
Okay.
yeah, I was muted.
Oh, okay.
Okay, yeah, it's not Turing. Turing requires at least two substances which interact. Diffusion-limited aggregation is basically precipitation, which leads to patterns. The simplest example of which everybody who lives in cold climates would be familiar with is frost.
Yeah.
Okay. Snowflakes is another example, but you have to be in a cold place for these things.
Yeah. So it is. Yeah, it's not like reaction diffusion. It's actually just kind of a single.
The hypothesis here is that the basic mechanism is diffusion-limited aggregation. Now, there's a very recent paper which, if you give me a minute, I can dig out, which is starting to become more sophisticated than that. The diffusion-limited aggregation may just be a first approximation.
Yeah.
So, let me see if I can find this thing. I don't know.
Yeah. So diffusion-limited aggregation is where you have like some particle in the middle of a field and you're throwing things at it and it sticks to that thing, and then it builds up over time as you have more things that go across at random and you build up the structure. It's kind of like a snowflake where it starts from a sort of a single point and then it grows out. And sometimes you can get some pretty impressive pattern formation through that process.
Bacteria grow that way.
Yeah.
And there are recent articles about condensates in cells.
Right. Oh, yes, condensates.
Yeah, so I don't know if any of that relates, but I grew some bacteria one time for a project, and they grow like kind of like a snowflake. They're anyway, in fractal theory, they're called diffusion-limited aggregate. I don't know.
Yeah. Yeah. So yeah, so these are the diatoms we're talking about. This is the mode of morphogenesis. You can have this process of diffusion-limited aggregation, which is where these structures build up through deposition. And, you don't need any special process. In Turing's model, you have these two compartments, which are reaction and diffusion. In this case, you just have diffusion, and that's what the primary driver of this. So, so this talks about the external form of crystals is prismatic and bounded by straight surfaces which cut each other at certain angles. But the same form is seen in skeletons of many of the protists, especially the flinty shells of the diatoms and radiolaria, which are a related sort of organismal structure. Their siliceous coverings lend themselves to mathematical determination just as well as the inorganic crystals. And this goes back to Haeckel in 1905. So this is, again, where you have these kind of silicate morphologies, and they have this different mode of morphogenesis. So this is an interesting form of morphogenesis here. There's also this type of, this is a paper that's related to this mechanism of branching morphogenesis inspired by diatom silica formation. So this is a paper. What was that?
That's the latest. This is the latest paper on this stuff.
This is, 2023. So 2023. So this is where they talk about silica formation as a form of branching morphogenesis. And so this is a potential mechanism. So the abstract here, it talks about the silica-based cell walls of diatoms, which we saw in the image. They're prime examples of genetically controlled species-specific mineral architectures. So again, this is this deposition of silicate, and this is driven by the expression of genes, and it's species-specific. So you get different deposition patterns in different species. It's not just a physical process, but there's this interaction between the physical aspect and the genetic aspect. The physical principles underlying morphogenesis of their hierarchically structured silica patterns are not understood, yet such insights could indicate novel routes toward synthesizing functional inorganic materials. Recent advances in imaging nascent diatom silica, so these are these structures that I showed, allowed rationalizing possible mechanisms of their pattern formation. So we can look at these images and sort of analyze the structure, the branching structures, the sort of the depositional structures, to get a sense of the pattern formation mechanisms. Here we combine theory and experiments on the model diatom, Thalasseorsera pseudonana, to put forward a minimal model of branched rib patterns. So there's this whole class of models of branching morphogenesis. And if you think about branching, you can think about something like the lung, where you have branching tubes that are, they kind of repeat down the structure of the lung. And so there are these signals that tell the system when to branch. And so we have this minimal model of branched rib patterns, which are actually a fundamental feature of the silica cell wall. So you saw in that shell that you had these sort of radial patterns that repeated across the body of the shell. That those are these branched rib patterns. And so physically you can build a minimal model. You don't have to worry about the gene expression or anything. You just build a model of those things, and you look at their frequency and you look at their sort of formation. So what are the physics behind this? So we quantitatively recapitulate the time course of rib pattern morphogenesis by accounting for silica biochemistry with autocatalytic formation of diffusible silica precursors, followed by conversion into solid silica. So this is this diffusion-limited aggregation, where you have these things that are diffusible, they're moving across the cell, and they're aggregating in place. So in Turing's model, this is sort of the diffusion part, but we don't have the reaction part. We just have the diffusion part. We're explaining everything through the diffusion part, where diffusion is being limited by something that thing that it's sticking to, or it stops its movement and it forms this structure. We propose that silica deposition releases an inhibitor that slows down upstream precursor conversion, thereby implementing a self-replicating reaction diffusion system featuring a non-classical Turing mechanism. So this actually is a two-compartment system reaction diffusion, because you're then adding in this conversion step with an inhibitor. So you're saying that these diffusible substances there's some control there, and so you have the two-compartment system. But this is actually a non-classical Turing mechanism, so it's kind of a special case of this sort of morphogenesis. The proposed mechanism highlights the role of geometrical cues for guided self-organization, rationalizing the instructive role for the single initial pattern seed known as primary silicification site. So the pattern seed is the thing that we start with, that seed, and then all of the diffusible factors kind of bind to that and form this pattern. And that's how you get the pattern that you get. The mechanism of branching morphogenesis that we characterize here is possibly generic and may apply also to other biological systems. So while it's species specific in terms of how the architecture is organized, this process is generic. So, we can say that there are a lot of generic physical processes across development, across morphogenesis. They don't, you have like variation across organisms, but you also have these processes that are very generic and allow you to sort of extract them and say that these are things that occur across the wide variety of organisms and variation. So we have this citation by Dick. This is a paper from 2021. This is with Dick and Joseph Seckback and another author. Diatom morphogenesis. This is from this volume here. So this is the link. This kind of goes over more generally about, this is actually a book on diatom morphogenesis. This goes over a number of topics. So there's some interesting papers here on sort of some of the puzzles in diatom morphogenesis, dealing with single cells to chains of cells. So you have these clonal chains. You have valve morphogenesis. You have geometric models of concentric and spiral areola patterns of centric diatoms. Diatom pore arrays, periodicities, and symmetries. A lot of different, something on buckling, a geometric and biophysical multi-scale feature of centric diatom valve morphogenesis. That's actually by Janice Pappas and Dick. And then another paper on buckling forces. There's a lot of interesting biophysics, I think, in diatoms. Dick introduced me to diatoms a couple years ago, of course, and we've been working with diatoms off and on. And I think if people are interested in diatoms, we should start working on those again. We haven't done a good diatom research area. We haven't done a good diatom research project in a while, so I think it would be really interesting to revisit. Then Dick says, I have electron micrographs with aberrant branching patterns that could be used to test this PNAS paper. Yeah, I'd like to know more about that. I don't know. Obviously, data's good. Show aberrant seeds in this theory. Morphogenesis of single cells not done. Yeah, so there's a lot of opportunity for looking at single cell morphogenesis here. And, I don't know how generalizable it is, but there are a lot of, there aren't really, I guess, a lot of models of single cell morphogenesis that are, especially for diatoms, because they're very unique in that sense. So a few notes on diffusion-limited aggregation. So this is something we talked about in the meeting. This is a classical biophysics model that you can use to model aggregation. So we talked about morphogenesis. We talked about reaction diffusion models of Turing. And the reaction part and the diffusion part are two separate components. So the reaction part, of course, is where you have a chemical reaction. And the diffusion part is this part, where you have a diffusion of particles across sort of a space. And in this case, you have diffusion-limited aggregation. So as I described in the meeting, you have some sort of stationary particle that sits in one place, and then you have particles moving randomly across a space. And those particles, if they hit the stationary particle, attach to it. And so it forms a cluster. And then that cluster grows because the particles are moving randomly and running into the stationary structure, or what is now a structure. So eventually you end up with large-scale patterns that aggregate just simply through diffusion and having some limit on that diffusion. And so this is a model by Wolfram Demonstrations Project. This is diffusion-limited aggregation, a real-time agent-based simulation. So wherever agent-based simulation, This is, of course, our particles that are going to move around, and we can modify the level of noise, so we can modify the stochasticity of the model. We can modify the number of particles. So let's start with probably about half of the number of particles if we can. Okay, there we go. And then we give it a random seed, so that's just any number we'd like. And then we can run the simulation by ticking this box. And so now the simulation's running, as you can see. We can put it full screen. So this is now our simulation. And as you can see in the middle, that red dot, that red structure, are these particles that are hitting. They're the stationary aspect of it. And so the particles are hitting, they're moving around randomly, but they're hitting this structure, and they're forming a larger structure. So we ran into some problems here. Let's reload this. Now we see a larger structure here. It's grown. It's hard to see. Let's run the simulation. You can see the structure starting to grow. And my level of noise is pretty low in this run, so you're not going to see a lot of aggregation. You can see here, these particles are moving around randomly. It's this sort of Brownian noise, but the level of noise is pretty low, so you're not getting a lot of aggregation. You're getting gradual aggregation. If we increase the level of noise, we start to get faster aggregation because the particles are moving around more, and their chances of bumping into the structure increase. And of course, as the structure grows, the chances of any particle being limited by that stationary structure also increases. So you can see we're starting to get some sort of pattern, some sort of branching morphogenesis here, where you get two branches. You can already see actually kind of three branches in this structure. And if we increase the level of noise some more, let's do that so we can speed things up. We start to get an increased accretion of some sort of branching morphogenesis. So you see this is simply through accretion. You have Brownian noise or some other type of noise. You have random movement of particles in a space. You have some stationary component, which you start with. It's your initial condition. And as things proceed through the simulation, as diffusion becomes limited by the structure, you not only get branching morphogenesis, but it starts to become easier to grow the structure. So you can see our structure is growing fast now. We're getting more and more particles stuck to this structure, and we're getting branching morphogenesis. Now we have like these 5 branches. So you can think of like budding and branching in a plant as maybe pursuing this kind of strategy. Now in plants, they don't actually do it this way because they're internal mechanisms. There's auxins, growth factors like that. We also have cells that are sort of branching out from the interior of the structure. But you can replicate plant growth this way. You can replicate bacterial growth in bacterial colonies, and you can replicate condensates, intracellular condensates. And so we can see here our structure. We already have 5 branches. We have more kind of coming up, and so this is what's going to happen. We're going to get a couple of maybe 2 or 3 branches, and then we're going to get sub-branching. And the whole structure is going to look fractal by the end of the simulation. It's going to have this almost like a snowflake, where you have intense branching patterns that are self-similar. And of course, if we run the simulation again and again, we get a self-similar pattern. We get a pattern that's different, but it's similar in the structure. So you could analyze it and say that it's part of the same class of structure, even though it's totally different in terms of the individual parts.
Well, yeah, there are about, I guesstimated 200,000 species of diatom. Okay. At least the taxonomy is clear enough. So
Yeah.
we've only published on 12,000.
Yeah.
But the general problem is there. It's in diatoms, it's in ciliates. Radiolarians also use silica, but they're entirely different from diatoms.
Yeah.
And desmoid. I mean, there are lots of single cells where there are patterns on or of the single cell, which shapes the cell itself, but patterns within the cell or on its surface, which are basically not understood.
Yeah.
Okay. And how far back this goes, I'm not sure. I know there are some weird bacteria also that have some morphogenesis, or even colonial multicellular groups of bacteria.
Yeah.
So it's basically an unexplored area.
Yeah.
Now the electron micrographs I have are by Ryan Drum, who unfortunately can't participate because he has dementia.
Oh, okay.
Okay. So he gave me these micrographs in the 1960s.
All right.
Okay.
Yeah.
So it's completely open. It requires some serious simulation. It could simulate the DLA model. It could simulate the model that you discussed, and see if it actually works. Because I have 100 of them.
Yeah. Oh wow. Okay.
And they're all different, and each one has branching patterns that look uniform but come off of an aberrant seed, what these people are calling seed.
Yeah.
Okay. So there's an opportunity here to try to simulate them with different models and see what model works. I mean, I tried in the paper that I did with Drum. We tried, but computers were too slow.
Yeah. Yeah. That.
Okay, but computers are much faster now. So,
Right.
At any rate, the simulation from the atomic level to the electron microscope level gives you an idea of what's going on, but it requires simulations that can move single molecules around in space and see what patterns develop.
Yeah.
I mean, even if you take the non-living world, I don't know. I haven't seen any simulations of snowflakes, for example.
Oh right.
Okay.
Yeah.
And and there's this hypothesis that every snowflake is different.
Yeah. Well, I think there's self-similarity, so they're not exactly the same, but they're very similar in the structure. I've seen DOA simulations online. Yeah. It's just that it's kind of a passe thing, I guess. People are not as fascinated as they once were with it, but there's still a lot of interesting stuff there.
Yes, I agree. At any rate, the diatoms are fascinating because they have so many different structures which are unexplained.
Okay.
Okay, if you go across different species.
Yeah.
Okay. And this individual variability with the aberrant diatoms is unexplored. Okay.
Yeah.
I'm waiting for a very strange thing, some special kind of paper that allows you to turn a scanner into a film digitizer. I don't want to buy a whole new machine for a film digitizer.
Right.
Okay. So as soon as I have that, I can then copy these micrographs and put them online.
Yeah.
Okay. But it might be a good thing for DevoWorm because if somebody's interested in getting into the single cell morphogenesis problem. And this new theory could be right, could be wrong. We don't know, but it's not tested.
Yeah.
So it's full of parameters which are speculations about what might be happening.
Right. Yeah. Okay. I think that's it for today. Thank you, Dick, for the kind of exploring that area. Oh, Susan?
Hi.
Hi.
Did you get my USB stick with the images from the microscope?
Well, how long ago is that? Like
Oh,
I've gotten
about
a couple.
month or two.
A month or two? Maybe. Yeah, I think so. But I think that was more than a month though.
Yeah,
Yeah.
I sent them a while ago.
Yeah, I did get that. Yeah. Yeah, I do have them. Yeah.
All right. Yeah. I just still want to know what you think of the quality of them because they're more blurred than I would like.
Yeah, they were a bit blurred, but I mean, it's like something that we have to kind of smooth out on and post processing. So sometimes it's not. But yeah, I mean,
I could try sticking them in MATLAB and doing some image processing on them, I guess.
Maybe, but yeah, I mean, I don't know. I don't know if you can get it any better. It's kind of a hard thing to.
Well, it's the quality of the microscope, I guess.
Well, yeah, I mean,
the depth of field. It's a depth of field thing more than anything.
Yeah. Okay. Well, yeah. Go ahead.
No, that's all. I just don't know how to find something with something greater than 3 megapixel resolution.
Oh.
Anyway,
yeah.
It's you'd have to have better lenses on the piece of equipment as well, and then the cost would go up. I don't know.
Right. Okay. Well, okay. Thanks for attending the meeting. Again, let's keep in touch over the week and see everyone next week.
Okay, great. I'll leave this open.
All right.
Okay,
Thanks. All right, everyone. Take care.
Goodbye, everyone.
Bye.
Bye.
Bye.